Welcome to today's lecture on cell biology, specifically focusing on protein-protein interaction networks and their significance in various cellular processes. Protein-protein interactions play a crucial role in signal transduction, gene regulation, and disease pathology. In this lecture, we will explore different aspects of protein-protein interactions, including their structural determination, post-translational modifications, computational prediction approaches, and their impact on protein localization and transport. Additionally, we will discuss the importance of targeting protein-protein interactions for drug discovery and design. Let's dive into the fascinating world of protein-protein interactions and unravel their complexity. Proteins are essential in cells, but many processes require multiple proteins working together. Protein-protein networks show the physical contacts between proteins, enabling cell functions. The yeast-2 hybrid system is a popular method to study these networks, revealing complex maps of interconnected processes. PTMs are chemical modifications that occur on proteins after synthesis. They regulate protein-protein interactions, like phosphorylation promoting/inhibiting interactions. PTMs like acetylation, methylation, and ubiquitination change protein conformation, stability, or binding affinity. PTMs can also create docking sites, allowing precise control over interactions and revealing regulatory mechanisms. In summary, X-ray crystallography is an important method for determining the structure of protein complexes. It helps reveal how proteins interact with each other and informs drug discovery. However, it has limitations in capturing dynamic interactions and requires well-ordered crystals. Signal transduction pathways are essential for coordinating cellular responses to external cues. Protein-protein interactions play a crucial role in these pathways by transmitting signals from the extracellular environment to intracellular components. They also regulate the localization and activity of signaling molecules, ensuring efficient signal propagation. Disruptions in these interactions can lead to diseases like cancer and neurodegenerative disorders. Protein-protein interactions are intimately involved in disease pathogenesis. Malfunctioning interactions contribute to various diseases, including cancer, Alzheimer's disease, and viral infections. Understanding the aberrant protein-protein interactions underlying these diseases is essential for developing targeted therapies. For instance, in cancer, oncogenic proteins often hijack normal protein-protein interactions to promote cell proliferation and survival. Therapeutic approaches aimed at disrupting these abnormal interactions hold promise in cancer treatment. Furthermore, studying viral protein-protein interactions provides insight into the mechanisms of viral replication and host pathogen interactions, aiding in the development of antiviral strategies. To quantitatively analyze protein-protein interactions, several techniques have been developed. One such technique is fluorescence resonance energy transfer, FRET, which measures the distance between two fluorophores attached to interacting proteins. FRET-based assays enable the monitoring of dynamic protein-protein interactions in real time and in living cells. Another quantitative method is isothermal titration calorimetry, ITC, which measures the heat released or absorbed during a protein-protein interaction. This technique provides valuable information about the thermodynamic parameters underlying protein complex formation, including binding affinity, stoichiometry, and enthalpy changes. These quantitative methods enhance our understanding of the kinetics and energetics of protein-protein interactions. Protein-protein interactions play a vital role in gene regulation. Transcription factors, for instance, engage in complex formation to orchestrate the precise expression of genes. Co-regulatory proteins interact with transcription factors to modulate their activity, either enhancing or inhibiting gene transcription. Additionally, protein-protein interactions are involved in chromatin remodeling and epigenetic modifications. Proteins that participate in these processes interact with each other to regulate DNA accessibility and alter chromatin structure. Dysregulation of these interactions can lead to aberrant gene expression patterns and contribute to developmental disorders and cancer. Protein-protein interactions are promising targets for drug discovery. Traditional inhibitors may not be effective, so new approaches are being developed. Peptidomimetics mimic protein interfaces to disrupt interactions, while small molecules can stabilize or destabilize complexes. Understanding these interactions helps design inhibitors for disease treatment. While many protein-protein interactions conform to the canonical model of direct physical contacts between proteins, emerging evidence suggests the existence of non-canonical interactions. These interactions involve additional factors, such as RNA molecules and other biomolecules. Non-canonical protein-protein interactions play crucial roles in various biological processes. For example, RNA-binding proteins can mediate interactions between RNA molecules and proteins, forming regulatory complexes involved in RNA processing and translation. Elucidating the mechanisms and functional significance of these non-canonical interactions enhances our understanding of complex cellular processes. Computational methods have become instrumental in predicting and analyzing protein-protein interactions. Using databases of protein structures and sequences, algorithms can identify potential protein-protein interaction partners based on complementary binding motifs or physicochemical properties. Machine learning algorithms trained on known protein-protein interactions can also predict new interactions and determine their structural features. Furthermore, computational modeling techniques aid in understanding the conformational changes and dynamics of protein complexes. These computational approaches complement experimental methods, enabling the generation of hypotheses and guiding experimental design, ultimately providing valuable insights into the mechanisms and functional implications of protein-protein interactions. Protein-protein interactions are not static entities but rather dynamic complexes that can change their composition and conformation in response to physiological or environmental cues. Dynamic interactions can involve the exchange of interacting partners within multi-protein complexes or the formation of transient interactions. Techniques such as nuclear magnetic resonance, NMR, spectroscopy and cryo-electron microscopy, cryo-EM, allow for the study of dynamic protein-protein interactions. These methods provide information about the flexibility, kinetics, and conformational changes occurring during complex formation. Understanding the dynamics of protein-protein interactions expands our knowledge of how these interactions regulate cellular processes. Protein-protein interactions are crucial for protein localization and transport in cells. Specific interactions with chaperones, adapter proteins, and transport machinery help proteins reach their intended destinations. Disrupting these interactions can cause protein mislocalization and dysfunction. Studying protein-protein interactions is important for understanding cellular organization and function. Protein-protein interactions are essential in cellular processes. Studying their networks, modifications, and structure helps us understand cellular functions. Quantitative and computational methods predict and analyze interactions, aiding in finding therapeutic targets. Dynamic interactions improve our understanding of cellular organization. Exploring these interactions opens doors for new therapies and drug discovery.